In this video, we will discuss about another approach or blueprint that is presented in the paper that talks about this enhanced anti-tumor efficacies of the T cells before reinfusion. So idea is more or less similar. They have just combined it into a formal representation. So what it is, in order to do that, first we need to have a tumor samples and also the healthy samples of the same patient. Then sequencing of those samples, tumor samples and the healthy samples will be done to find out the point of mutations, and thereafter a mini genes will be created or synthesized that contains the mutation informations, and these mini genes will be inserted into the plasmid, and thereafter the plasmid will be electroporated into the autologous antigen presenting cells, and these antigen presenting cells will then be able to translate those mini genes into the RNAs, and they can express these RNAs into the major histocompatibility complex, and thereafter this co-culturing of these APCs with the T cells will help in transferring those mutated RNA informations to the T cells. That is shown in the figure here. The major histocompatibility complex transfer the mutated RNA informations to the T cell receptors, and this will help the activations of these T cells, and those T cells that are activated by this process will be screened through the activation markers, and some of the prominent activation markers are these CD8 plus positive cells, which represent this 41BB activation, or OX40, or which represents the CD4 plus T cells. And once these activated T cells are been sorted, these T cells will be expanded, and these T cells will not only have the informations about the tumors, since they come from the tumor infiltrating lymphocytes, but so in this case they have used the T cells from the blood or the tumor, so it can either comes from the tumors or from the blood. It can be from the tumor infiltrating lymphocytes or from the blood itself. So if it is from the blood, then they will have the informations to target those mutations that are present within the patient. So these expanded T cells will then be reinfused into the patient after this lymphodepletion, and we have discussed many times in our previous videos why this lymphodepletion is needed and what are the problems that might occur, and sometimes that problems might even leads to a fatal conditions or lethal conditions, because if those side effects such as cytokine release syndrome crosses the threshold that patient can tolerate. So this is a blueprint that can be used to not only increase the efficacies of the anti-tumor, but make it more specific or customized based on the patient, to target only those mutations that are present within the patient. As sometimes because of the universal target antigens, what will happen is that the efficacies of those T cells is not that good, because there are mutations that are present within the patient that does not that cannot be accounted by the universal T cells, universal antigen T cells that are commercially been sold out. So in order to make sure that the tumor's regressions occurs to its maximum possible extent, so it is best to have the T cells that are activated and have the informations about the mutations that are present within the patient, against which this T cells is going to be reinfused. So this is just a small blueprint or a brief blueprint of how to increase the anti-tumor efficacies or customize these based on the mutations that are present within the patient. If you need to understand more about this idea in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.